Hello everyone, welcome to our channel Biological Concepts. In today's video, we will discuss some important questions on GATE Xl Microbiology 2025. Some brief notes regarding the questions and their related options are also provided here, which will be beneficial for you. So stay tuned in this video. First question, which of the following metabolites is associated with bacterial stringent response? Option A, cyclic di-GMP, that is CDG. Option B, guanosine tetraphosphate, that is PPGPP. Option C, cyclic AMP, that is CAMP. Option D, cyclic GMP, that is CGMP. The correct answer is option B, guanosine tetraphosphate. The guanosine tetraphosphate, often referred to as an alarmon. It is the key signaling molecule that is synthesized in bacteria during the stringent response. Now, what is stringent response? The stringent response is bacterial survival strategy triggered by nutrient deprivation or other stressors, which leads to changes in gene expression and metabolism to conserve resources. The guanosine tetraphosphate is produced by enzymes like REL-A and SPO-T. It acts by regulating replication, transcription and translation process to help bacteria to adapt to stressful condition. It specifically upregulates stress response genes and downregulates process like ribosomal RNA synthesis, which is essential for growth. Next question. India is aiming to be free of tuberculosis by 2025. One of the key approaches for this program is DOTS. Which one of the following options is the full form of DOTS? Option A, directly observed therapy short course. Option B, directly observed tuberculosis short course. Option C, district operated therapy system. Option D, directly operated therapy short course. The correct answer is option A, directly observed therapy short course. DOTS stands for directly observed treatment short course. It is a strategy which is recommended by World Health Organization for tuberculosis control. Nowadays, drug resistance due to tuberculosis is a big problem. This approach involves healthcare workers directly observing patients taking their medication to ensure their complete full course of treatment, which helps prevent the drug resistance. The key components of DOT strategy. One is political component with increased and sustained financing. Second is case detection through quality assured bacteriology. Third, standardized treatment regimen with direct observation of treatment. Fourth is effective drug supply and management system. Fifth is monitoring and evaluating system and impact measurement. Next question. Correctly match the bacterial type in column 1 with their corresponding environmental niche in column 2. In column 1 is psychrophile. Q is barophile, R is bacterial type, this is mesophile, S is halophile. In column 2, 1 is pressure greater than 380 atmosphere. 2 is temperature between 15 degrees centigrade and 45 degrees centigrade. 3 is temperature below 15 degrees centigrade. 4 is pH less than 3. 5 is salt concentration greater than 2 molar. So, here option A, B, C, D present and you have to choose the correct option. So, psychrophile, we know that psychrophile are bacteria that thrive temperature below 15 degrees centigrade. So, option P is, so P, that is psychrophile P is related to 3. So, P is 3. Here in P is 3. In option B, P is 2, it is not correct. Option C, P is 1, which is also not correct. And option D, P is 5. That is, P is 5 means salt concentration greater than 2 molar. Psychrophile, not. Psychrophile is the bacteria that thrive temperature below 15 degrees centigrade. So from this only one option you can choose that the option A is correct. And let's see another option, that is Q, barophile. Barophile is an organism that thrive pressure greater than 380 atmosphere. Third is R is mesophile. Mesophile is the bacteria that thrive temperature between 15 degrees centigrade and 45 degrees centigrade. That is, R is equal to 2. S, halophile. Halophile is a bacteria that thrives at salt concentration greater than 2 molar. That is, S is 5. So option A is correct here. Okay. Now, psychrophiles are bacteria that thrive in cold temperatures, specifically below 15 degrees centigrade. Examples are Arthrobacter species, Psychrobacter species. Barophiles, also known as pisophiles. Baro means pressure. So these are the bacteria that can survive and thrive under extremely high pressure greater than 380 atmosphere. Examples include Morchiella, Chlorella, Photobacterium, etc. Next is mesophiles. Mesophiles are organisms that prefer moderate temperatures. Meso means moderate, typically between 20 degrees centigrade and 45 degrees centigrade, with an optimum temperature around 37 degrees centigrade. Next one is halophile. Halo means saline. So these are bacteria that require high salt concentration or saline concentration for growth, specifically more than 2 molar. Whereas if you see that this option that pH less than 3, the bacteria which thrive in pH less than 3 are known as acidophiles. Okay. So these are the options. Okay. Next question is, Robert Koch used a meat-infused nutrient medium for which one of the following purposes? Option A, to grow disease-causing microorganisms. Option B, to demonstrate presence of microorganisms in air. Option C, to test the efficacy of sterilization approaches. Option D, to demonstrate antimicrobial activity of soil isolates. And the correct answer is option A, to grow disease-causing microorganisms. Koch's contribution to microbiology focused on identifying the specific microorganism that cause specific disease. A key part of his method is using meat-infused media, which allow to cultivate these pathogens in a controlled lab environment, making them available for study and identification. How the microbes grew and behaved on this meat medium. Koch successfully isolated and identified causative agents of disease such as anthrax and tuberculosis. So, Robert Koch used meat-infused nutrient medium to grow the disease-causing microorganisms. So, this option A is correct. Next question. A bacteria obtain energy from a chemical source by the oxidation of reduced nitrite ion with carbon dioxide as the principal carbon source. Which one of the following nutritional groups does the bacterium belongs to? Option A, photoautotrophs. Option B, photoheterotroph. Option C, chemoautotroph. Option D, chemoheterotroph. Photo means light, that is those who require light as energy source. Here it is mentioned that they obtains energy from the oxidation of reduced nitrites, that is reduce chemical compounds. So this must be chemo, and use carbon dioxide as the sole principal carbon source. Those organisms which use carbon dioxide as principal carbon source is known as autotrophs. So the option will be chemoautotroph. This is the correct answer. Chemoautotroph is the correct answer. Let's discuss chemoautotroph. Organisms that obtain energy from chemical reaction are known as chemotropes. Okay, next is autotrophs, organisms that can synthesize their own food from inorganic compounds such as carbon dioxide. So here this they use obtain energy from chemical source by oxidation of reduced nitrite. So they are chemotrophs. Again, they use carbon dioxide as their principal carbon source. So they synthesize their own food from inorganic compounds, from this inorganic carbon dioxide. Okay. Now, chemoautotrophs are a specific type of chemotrophs that are also known as autotrophs, meaning they obtain both energy and carbon from inorganic sources, such as carbon dioxide as their principal carbon source and use reduced nitrite as their chemical source. Some nitrite oxidation, some bacteria can oxidize nitrite to nitrate and gain energy in the process. Carbon dioxide as a carbon source. The bacterium uses carbon dioxide as a carbon source, which is the defining characteristics of autotrophs. So this option is correct, that is chemoautotroph. Now let's discuss another options. That is phototrophs. Photo means light. That is, these organisms use light as their principal energy source for metabolism. Heterotrophs, this is an organism that cannot produce their own food, instead taking nutrition from other sources of organic compound, organic carbon, mainly matter from other organisms. Photoheterotrophs, this is an organism that depends on light for most of its energy and principally on organic compounds for its carbon. These are photoheterotrophs. Okay, those that use light as their energy source and organic compounds for its carbon are known as photoheterotrophs. Now, chemoheterotrophs. Chemoheterotrophs are organisms that get both their energy and carbon from organic compounds by consuming other organisms or their waste products. The chemo part indicates that energy comes from chemical reactions, and hetero part signifies they cannot produce their own organic compounds and must obtain them from external sources. Examples include most animals, fungi, and many bacteria, which are crucial decomposers that recycle organic matter in ecosystem. Next is chemoorganotrophs. Is an organisms that obtain energy from oxidation of reduced organic compounds. Next is chemolithotrophs. These are organisms that are able to use inorganic reduced compounds as energy source. Litho means that use inorganic compounds as their energy source. Next question. Which of the following sites are the locations of ATP generation through oxidative phosphorylation in E. coli? Option A, inner membrane only. Option B, outer membrane only. Option C, both outer membrane and inner membrane. Option D is mesosome. And the correct answer is option A, inner membrane only. Now let's discuss why inner membrane is the location for ATP generation through oxidative phosphorylation in E. coli. In E. coli, which is a prokaryote, does not have mitochondria. The oxidative phosphorylation, the process of ATP generation via electron transport chain, takes place in the inner membrane. This inner membrane, also known as cytoplasmic membrane and plasma membrane. So we know that the prokaryote does not have cell membrane bound organelle, thus they does not have mitochondria, which is basically the ATP generation process in most eukaryotes. Here in case of E. coli, the process of ATP generation occurs takes place in the inner membrane, that is the cytoplasmic membrane or plasma membrane. In the E. coli, they does have outer membrane that primarily function as protective barrier containing porins, but it does not house the components of electron transport chain or ATP synthase. And mesosomes are the invagination of the plasma membrane found in prokaryotic cells, but are now considered artifacts from chemical fixation rather than real structure in living cells. So they are not involved in oxidative phosphorylation. So in case of E. coli, the inner membrane is the site for ATP generation via electron transport chain. Next question. The adaptive immunoresponse in an animal involves the generation of antibodies against an invading bacterial pathogens. The following graph represents antibody titer levels in a mammal exposed twice to the pathogen. Which of the following options correctly pairs antibodies to peak 1 and peak 2 in the graph? This is the peak 1 and this is peak 2 in the graph. This is the first exposure of the pathogen. This is the second exposure of the pathogen. X-axis represents the time, whereas Y-axis represents the antibody titer level. Option A, peak 1 is IgG, peak 2 is IgM. Option B, peak 1 is IgM, peak 2 is IgG. Option C, peak 1 is IgE, peak 2 is IgG. In primary response, we know that the IgM appears first, and in secondary response, IgG appears. After the initial exposure to the pathogen first time, the adaptive immune system begins to develop antibodies. Here IgM predominates. This process takes time, so the antibody levels are initially low and rise gradually, followed by a decline. Like that. Increase followed by a decline. Okay. When the animal is exposed, that is the bacteria is exposed to the same pathogen, second exposure, the immune system remembers it. This memory allows for a faster and more effective response. During second exposure, a rapid and steep increase in the antibody levels, that is the in secondary exposure, the IgG level predominates, reaching a higher peak concentration than seen during the first exposure. The antibodies may also stay at a higher level for longer. Here peak is like that. Okay, that's why the option B is correct, that peak 1 IgM and peak 2 is IgG, where peak is higher. Next question. Correctly match the viruses listed in column 1 with the nature of their corresponding genetic material listed in column 2. In column 1, P is bacteriophage lambda, Q is bacteriophage M13, R is coronavirus, S is reovirus. In column 2, 1 is double-stranded DNA, 2 is single-stranded DNA, 3 is single-stranded RNA, and 4 is double-stranded RNA. Here option A, B, C, D is present, and you have to choose the correct option. Now, as you know that bacteriophage lambda has double-stranded DNA and bacteriophage M13 possesses single-stranded DNA. So Q is 2, P is 1, and coronavirus has single-stranded RNA and reovirus possesses double-stranded RNA. So S is 4. So where the option P is 1, Q is 2, R is 3, and S is 4. In option C, that is P is 1, that is bacteriophage lambda has double-stranded DNA, Q is 2, that is bacteriophage M30 and single-stranded DNA, coronavirus, option 4, 4, coronavirus has single-stranded RNA, that is 4 is 3, and S is reovirus, reovirus has double-stranded RNA, that is S is 4. So option C is correct. Reovirus utilize single-stranded RNA as an intermediate during their replication cycle, although their genome is composed of double-stranded RNA. So reovirus genome is double-stranded RNA. Coronavirus is a spherical encapsulated positive sense single-stranded RNA virus. So coronavirus is single-stranded RNA virus. Bacteriophage M13 single-stranded DNA virus. Bacteriophage lambda, it contains phage double-stranded linear DNA genome. So phage lambda has a double-stranded DNA. Next question. Which option correctly matches the structure of bacterial cell in column 1 with their corresponding function column 2? In column 1, P is cell wall, Q is fimbriae, R is flagella, S is pili. In column 2, the functions are like that. One is protection from osmotic stress, 2 is attachment to surfaces, 3 is motility, 4 is transfer of genetic material. Now you have to choose the correct options. Here option A, B, C, D is present, and you have to choose the correct option. Protection from osmotic stress, we know that cell walls helps in protection from osmotic stress. So P is 1. Attachment to surface, fimbriae helps in the attachment to surfaces, flagella helps in motility, so R is 3, and pili transfer helps in transfer of genetic material, S is 4. So in option A, P is 1, Q is 2, R is 3, S is 4. So option A is correct. So here option A is the correct option. Okay. Now let's discuss the function of cell wall, fimbriae, pili and pili. Okay. Now function of cell wall. The cell wall serves several key functions. One is it provides structural support and helps maintain cell shape. Second is it acts as a protective barrier against mechanical damage and pathogens. Third is it prevents osmotic bursting by regulating water intake. That is, it prevents from osmotic stress. 4 is additionally it facilitates cells to cell communication and regulates the passage of materials into and out of the cell. Next, function of fimbriae. It primarily functions in bacterial adhesion, enabling bacteria to attach to surfaces, host tissue and other bacteria. So it helps in attachment. Second is these short hair-like structures made of pilin proteins are essential for forming biofilms and colonize environment. Third, they also play a key role in pathogenicity by helping bacteria adhere to host cells, resist removal mechanisms like flushing, and evade the immunodefenses. Next, function of flagella. The main function of flagella is to provide motility. So the function of flagella is provide motility and enable cells to move through the liquid environment. Beyond locomotion, flagella also serves as a sensory organelle to detect environmental changes like pH and temperature. The flagella can facilitate virulence by helping bacteria to colonize surfaces and play a role in cell adhesion and biofilm formation. Next, function of pili. Pili in bacteria have two primary functions. So, the adhesion to host surfaces which aid colonization and infection, and DNA transfer through a process called conjugation. So they helps in colonization, also in conjugation. This conjugation is the form of bacterial sex and promotes genetic diversity. A special type of pillars known as sex pillars facilitates the transfer of DNA between bacteria, providing genes for antibiotic resistance. Thank you. If you like this video, please like, share and subscribe to this channel. Thank you.